Welcome to The Joe Cohen Show. Join me as I share my experience with biohacking and invite top health experts to explore the latest technology, supplements, research, and resources for optimizing your body and brain. I will be able to, what I would like to do is show it to you, and you're going to tell me, hey, look, this is what's going on. Here's what you should do. And I'm going to do it. It's not going to be a client that you would have that's going to do things as much as me. I will do, okay, whatever. I'll take 100. I already take 130 supplements a day. What do you want me to do? Take this one out? All right, I also know what each one's doing. And it's whatever you tell me to add. I'll take 150, whatever it is. Like, I'll do it and then recheck and verify that if what I'm doing is actually changing a certain marker. But one of my big concerns about this, and it's going a little out of my framework, because I have a very specific framework now. My framework, and it works for me. So I've achieved amazing results, and I've seen other people achieve a great result. The framework is as follows, right? I try to work with the information that is most scientifically proven first, right? And I think that makes the most sense to start with. And so I look at these markers that have all-cause mortality data. And there's also metabolomic studies that show different metabolites in the blood usually. If this metabolite is higher, it's associated with longevity or some metric, some positive metric. And so that's why I actually started doing amino acid tests, because there were a bunch of metabolites there, amino acids, that were associated with longevity. And I said, okay, let me try this out. And I started taking them 10 times already. Just boom. Okay, let me add this in. And actually, all of them changed. I was able to change every single marker pretty much. Be like, okay, I want to increase this. Let me do this. And it did it. Like, my histidine was low and arginine was low. Okay, let me take arginine and histidine. Increased, right? What do you know? Boom, increased. And some of them, I'm not going to go through all my results exactly, but the idea, just because that's not the place for it. I've done it before, but the idea is that I can take a marker and I could develop a hypothesis about, okay, let's take this and see what changes. And I will see better results often when I optimize these markers. And there's a bunch of these markers that are related to all-cause mortality. My biggest concern with these tests, and by the way, I think you're a very credible figure, so I appreciate all of your opinions, and I plan on testing all of your hypotheses out, believe me. But my biggest concern is that I'm not seeing the data for all-cause mortality for almost all of these markers, and especially for the urine, right? So if I do see it, I'm generally seeing studies with serum metabolomic markers, not urine ones. And my question is, why these tests have been out for a really long time? Why aren't researchers not looking at this data? Like, even if they're looking at kynurenine and mortality or something like that, I've seen certain studies related to kynurenine in the blood. They're looking at serum. They're not looking at urine. So my concern is, like, my approach until now has been follow the research. And even if it's preliminary, these metabolomic studies are considered very preliminary, but still I've had good results, right? Because just getting things in the optimal range seems to work, right? It seems to overall have a positive impact. Why are we not seeing this for these urine tests?
That's a good question. I'm not
Where are
sure,
we?
right? Because if you look at the organic acids, and to answer your question, what we really need to ask is, what is an organic acid, right? And if you look at what an organic acid is, essentially, if you look at biochemistry, with biochemistry, you need a substrate, and then you need an enzyme to convert that substrate into something, and that enzyme needs a cofactor to do its work, right? And so in a way, and then the end, I guess the metabolites that are being produced through these biochemical functions is what an organic acid is. And so what they're measuring on something like the organic acid test is, if something goes wrong, if the biochemistry doesn't work optimally, then either they're going to dump the substrate in their urine, or the metabolic pathway is going to take an alternate route and it's dumping an alternate metabolite in the urine, or there's less of the intended metabolite being produced. So there's less of that metabolite dumped into the urine. And that's what an organic acid test measures. So it actually very similar to the amino acid, urine amino acids is it actually gives very, very good information on biochemical patterns. And for me, I guess if I was talking longevity, and to be honest, Joe, I haven't really I haven't really made it my work to check a lot of research in terms of like how research like views a lot of these. I look more at when I researched the organic acid test, like I looked at the metabolic pathways because I wanted to figure out what makes glyceric acid high, what's make glycolic acid high, what would be changing these markers. So that was what I was interested in. And if I was going to say, let's say something, let's take the neurotransmitter metabolites. And like I said, the dopac is not on here. But if you would expect dopamine to be metabolized as HVA. But if dopac was really high and HVA was very low, then that would tell me that dopamine metabolism is not happening as it should. So that it would mean that the person is ending up with a lot of dopamine metabolite. And dopamine metabolites are toxic to the brain. They kill dopaminergic neurons. They will set you up for Parkinson's disease or other kind of neurological issues where dopamine neurons become damaged. So for me, I don't even need a research paper to tell me that this is definitely not something that you want to happen if you are interested in longevity. For me, that's just one example. If you had, I guess, another reason would be the uracil and the thymine, right? Because thymine is essentially methylated uracil. So if uracil was very high, thymine was very low, then we know that there's more RNA molecules than DNA molecules being produced. That means there's more uracil potentially being inserted into DNA, making the DNA more fragile, which then sets you up for DNA damage, chronic illnesses, all of those kinds of things moving forward. So for me, there's a lot of markers on here that I could link to healthy aging or all of that kind of stuff. And I don't know why it's not that mainstream. But what I have seen is, at least here in Australia and in a lot of the hospitals, is often they don't use these specific organic acid tests. Like this is the Great Plains one that I've got here as an example. They've got their own labs, but often they will use that in the hospitals as a last resort. When they've done all the testing in the world and they still can't figure out what's wrong with someone, they will start measuring for organic acid. Not necessarily the exact same ones here, but for some kind of organic acid. So they do
In the
use it.
serum or urine?
I believe serum, so not urine, right? And so then it does, bring up the point that you've made earlier is, okay, how does it differ? How does serum and urine differ? And they would differ because same as with amino acids, serum amino acids and urine amino acids, I don't think that they will necessarily correlate, although I've never really done that experiment, but I wouldn't expect them to be exactly the same.
So what I've seen, when I come to my conclusion, like the reason I started testing these serum amino acids is because I started to see research, sometimes Mendelian randomization research, sometimes other metabolomic research, higher levels of this in the serum. And problem with these tests, and again, I think you've explained quite well the biochemistry and theory of why different things would make sense. And it's enough for me to test the theories that you have, essentially. Like, I'm willing to, if nobody could have any theories whatsoever that made any kind of sense, I would just not even bother with it. But the idea is that, first of all, it's two steps removed because the metabolite from the serum and the urine, all of a sudden you introduce a lot of complexities, right? You're urinating out something a lot more, which could mean maybe you're taking it in a lot more, right? There's a way more complexities once you're going from serum to the urine rather than checking directly the serum. It would make a lot more sense for me to check the serum directly if you want a more direct response of what your body is doing. It's, okay, this is high in the blood, therefore it would make sense that this means that, right? You would start looking at the biochemistry. It's not always going to,
I would
yeah,
challenge you on that. I'd say ideally you'd want to do both. You'd want to do serum and urine, and then you want to compare
I
them,
would
right?
agree with
Because
that.
even with
Yeah.
serum doesn't mean it's getting into the cell. It just means it's in the bloodstream, right? And you could have high levels in the serum and very little levels in the cell. And we see that a lot, right? We see that a lot I personally see it a lot.
But
Whereas with
how would
serum,
that change anyway? How would that change the urine? So let's say you have high in the serum, how would low or high in the urine impact what's going in the cell?
It depends
If
on what
something's
you're
high in the
measuring.
serum,
It would depend on what you're measuring, right? So I agree with you. If you're taking something high in the serum and it's not absorbing, then you'd expect that you would just urinate it out, right? So high in the serum, high in the urine kind of thing. But if you're measuring what the let's use amino acids as an example here. If you're measuring whether the amino acids is actually being used in a certain way, then you'd be wanting to see if it gets into the cell. And let's use an example of I don't know, tyrosine, right? So tyrosine has to be metabolized into it gets used for thyroid hormone. Tyrosine is also used to obviously make dopamine and norepinephrine and all of that. So let's say your tyrosine you're taking tyrosine, and your tyrosine is like really high in the serum, and it's also high in the urine. But let's say, I don't know, let's say your dopamine metabolites or dopamine is low and norepinephrine is low and T4 is low. Then you'd have to question, is the body actually using that tyrosine? Am I maybe missing the cofactors, which could be minerals or vitamins or what have you, and using that amino acid? I guess for me,
yeah, but I'm not sure that would influence you. I agree with you, by the way, right? If you're having high levels of something, then you're going to start questioning, wait, am I utilizing this thing enough, right?
yeah,
Which has happened to me in the past as well, meaning I found that certain nutrients, if my B vitamins are too high, for example, either I could be taking too much of it, or I'm just not, and even if I'm taking a lot of it, which I am, I'm still not utilizing all the amount that I'm taking. And so it's saturating the blood. The rest is being urinated out. I don't really think that it's the cells aren't being using it. I just think it's maybe not being utilized in a specific pathway because it doesn't have another cofactor. I would frame it a little differently instead of the cells using it versus it's not being utilized in a specific way because there's another rate limiting factor that has met that it has come up against, if you will.
yeah, no, that's a good point. I guess the other thing that I would bring up, and I don't really have any data on amino acids per se, but the other thing that you'd have to consider is also the half life, right? So I'm going to use histamine as an example. And so with histamine, I find that serum histamine is not a very accurate marker. And one of the reasons is the research shows that it's got a very short half life. Look, it could be 60 seconds or 90 seconds or something like that. And I've seen so many people with histamine issues and their serum histamine levels just come back normal. So a more accurate marker for histamine is it N-methylhistamine? I can't remember the exact one, but there's a urinary histamine metabolite that's considered a lot more accurate than serum histamine. So I guess that would be another thing that you just have to consider, depending on what it is that you're measuring, is okay,
That
what
would
is
make
the
sense. Would make sense. So, but I think it's more of the exception. Look, the more data you have, let's be clear about that. The more data you have, the better it's going to be. It could make it a little more complex. You have to spend a little more time analyzing it. But I'm a fan of just getting more data. I would agree that I would like to see both urine and serum, right? But again, the question, I don't know if there's an answer to this, but the question is, why are researchers not looking at it? Meaning they're not looking at urine for various outcomes, which you want to look at because whatever is causing something, right? You could say that if you have a certain kind of infection, it's going to lower your LDL and whatever. You could have all these theories and whatnot, but you want to look at, okay, what is the LDL number that is related to lowest all-cause mortality, right? And at what age? And that actually changes based on the age. For various reasons, older people actually do better with higher levels. But you also know that younger people can live longer if they have lower levels because there's less atherosclerosis. But the thing is that with the body, there's so much noise and there's so much complication that I like to look at what's the bottom line, right? All cause mortality or some kind of, at least it's like cardiovascular or big killer that if you've got higher levels of this or lower. And so you mentioned, okay, if you have high uracil and low cytidine or what was it? It was
timing.
uh,
Yeah.
thymine. Okay, you're like that. That's an indication of folate deficiency. But when they do studies, they find that you can actually just look at serum folate, and there's studies on all-cause mortality and serum folate, and they find that higher serum folate is associated with lower all-cause mortality, right? And so,
Mhm.
like, I just don't I agree, like, everything you tell me makes sense, which means that I think you're very smart and you have a good ability to analyze. I don't think you're dogmatic either, which is a great thing. So it's like, I like to learn things, but the question is, and I'm going to do all these experiments, just to be clear. The question is, why are they not researching these urine tests for all cause mortality data? That's what's the mystery to me. And I don't know if there's any answer, right? Like maybe they just haven't gotten around to it, but these have been around for a while and a lot of people are testing it. I don't know. It's just, yeah, it's odd.
I agree. And I wouldn't know. I could say that from my experience, mainstream medicine science can sometimes be like 20 years behind. That could be an explanation. But as you say, it's been around for a very long time. Yeah,
And they're
I
testing a
don't
whole
know.
bunch of other stuff, right?
Mm,
Like, you see a whole bunch of metabolomic studies coming out, microbiome. I think there's a huge delay between what happens in mainstream medicine and what comes out in the science. So, which is where people make big mistakes, is they think, oh, if it's not in mainstream medicine, then it's not scientific, which actually is quite not true. There's a lot of it's just not as standardized maybe, or right, it's not as validated. So you're not able to see, for example, very clearly in certain amino acids, it's not necessarily, they don't know if it's causal in every situation. You have to then use some of your own knowledge and analysis and to try to come up with a hypothesis. But at least I have that kind of big level data. Like people who lived longer had higher levels of this amino acid. Okay, very interesting. So if I've got lower levels, let me try to see what that's from and let me see if I can manipulate it. And then also, when I manipulate it, let me see what the effects are. So far, every time, not every time, but most of the times that I've manipulated these things based on the theories and based on the research, there's been very positive results. For example, I was low on histidine. I increased histidine. It turns out that I was also low on histamine because when I increased histidine, it just gave me a surge of histamine, which actually doesn't cause any of the effects that you would think that are associated with high histamine. Actually, I only got the positive ones. For example, I was basically like, one of my goals were, for example, more wakefulness, more energy. Histamine is important for wakefulness. It was like, I always felt like my gut motility was just not as fast, right? And actually, it's interesting because I took resistant starch over the past 11 years, right? I have my own brand, whatever, Joe's Resistant Starch, but I took it over the past 11 years. And I noticed that in the previous years, it took 20 hours to get from my gut to my large intestine because I felt the effect when it occurred. And now it's about 10 hours. So the motility has definitely improved quite significantly. And then I've done a lot of things to improve that. One of the things is actually histidine, which histamine increases the motility and the acidity of the gut. And I felt like my gut acidity levels actually just completely improved after that. Like, I felt like I just feel like my gut's not acidic enough. And we speak about histamine as like this bad thing. You say, "Hey, there's a lot of good things." I'm like, "Yeah, I was thinking I actually experienced the good things from histamine, from increasing it without." And what the research found is that when you take histidine, you don't have the negative effects from histamine,
no,
even though the
no.
histamine is increasing.
That's right. And I'll tell you something, and I'm glad you brought that up. So I see a lot of people who have, let's say, mast cell issues or histamine symptoms, right? The allergies and all of that. And when we do amino acids, in my case, I just do urinary amino acids, they have very low histidine, right? And this is my theory. My theory is that if people have had mast cell issues for a long period of time, they can eventually run short of histidine because the mast cells are producing all of this histamine. But that can then mean that you end up with a histidine deficiency that now the histaminergic neurons in both the gut and the brain don't have enough histidine to make histamine for gut function and for brain
Mhm.
function, right? And I agree
Right.
with you 100%. Like in those cases, when I've given those people, even though may have mast cell issues or what have you, we give them histidine, the mast cell stuff doesn't get worse. It's not now that their mast cells are producing more histamine, but the neurological symptoms get a lot better. So I 100% agree with you. I see the same thing in my clients as well.
Yeah, that's very interesting. Basically, your body's just going to be using a lot of histamine in other places. And It doesn't mean that, and you're probably becoming deficient in histidine, and you just need to replenish that, and it's not going to increase your histamine in the places it's already high or whatever it is, right? Like it's already
Yeah.
done its thing. It's not fueled. You would think it's fuel to the fire, but it's not. It's just like you already used the fuel, and then you need the fire to stay warm at that point.
That's right. And that's why you can actually get both high and low histamine symptoms at the same time, right? And I know it sounds like a contradiction, but the same with dopamine. You can get, I see it so often where people can get both the high and the low symptoms, and there's various reasons for it. That's also why nothing is black and white. It's not as easy as maybe we want it to be. Like things can sometimes be a little bit more complex, but that's a really good example of where you can have both high and low symptoms. Not unitarily, but a lot of people can have both of them at the same time.
So the last topic that I would like to discuss is genetic testing. Where do you see genetic testing fit into all this stuff?
Yes, I guess for me, I like to, because there's different types of genetic testing, as you know, Joe. Like some people focus on testing for disease genes, whether that be they're looking for specific genes like linked to disease. And then you obviously you have the gene that's not necessarily linked to diseases per se, but more the functionality, right? So if we take dopamine as example, you can test for dopamine production genes, dopamine breakdown genes, dopamine receptor genes, and it's more about giving you a picture of, okay, how is dopamine genetically functioning in this person? I tend to prefer or look more in that type of genetics because I'm more interested in what is this person's weak spots and strengths, what have you. I find that genetics, it's great. A lot of people like doing it because it gives them like a roadmap. Your genes don't change. So you got your roadmap and you can make dietary changes and lifestyle changes to keep yourself healthy, optimization of health. But I also will say that I see a lot of people, I'm going to use MTHFR as an example, a lot of people with MTHR SNPs, polymorphisms, and yet they don't show that when we do testing, they don't show that they're expressing MTHFR, right? The folate is great, the uracil thiamine is great, all of that kind of stuff. And then I see people who do not have any MTHFR SNPs, and they may have bad folate levels or what have you. For me, and this is what I explain to people, it's genes are like 10%, is like the other 90%. What are you exposed to? What's your diet like? What's your environmental exposure, stress, all of those kinds of things. But I do at the same time, when I take people's genetic tests and I explain to them what it means genetically, a lot of people will say, my gosh, that's me to a T. That's exactly, I do tend to have this mood or what have you, these kinds of like weak spots. I do use the genetics. I think it's going to be certainly, and we're probably already there. I think definitely I see now a lot of things in terms of what's the best medications to use in terms of your genetics, or what medications shouldn't you be using in terms of your genetics. Or they're now doing it with a lot of cancer tumors where they're testing for certain genes, I believe, and to try and discover what's the best treatment option for that cancer or that specific tumor. I think it's just going to keep increasing, that there's just so much research going into those areas. What I would hope for, where my caution would be, is that we don't start ignoring everything else. Like one of the main things that I really dislike is when people say, oh, we found the gene for autism or something like that. And I'd be like, there's no gene for autism. That's just ridiculous, because autism is a complex thing, right? It's more than just one gene. So I get cautious when people start talking like that. And that's, to me, that's just ridiculous. And it's ignoring everything around it and trying to minimize it to a gene. So I just hope that we don't go down that road and that we just use genetics as another tool, and forget about everything else per se.
I share your sentiments quite a lot. And even though I have a genetics company, I also think that the way that the functional medicine world currently uses genetics is not correct. Just, I'm speaking in a broad stroke, so I'm not saying you specifically, but in general, what I see, and you can, by the way, you can unshare your screen. I might just show you a few things, but yeah, essentially, the way the functional world uses genetics is they're looking at single variants, right? And you've
Hmm.
seen a bunch of these tests where they're looking at specific variants, and they're usually looking at like 100 variants or maybe 200, and they give you a list of SNPs and they color it. in different ways, right? That's what a lot of these functional tests that you see. The problem with that is that the research is pretty clear that there's all of your SNPs do something pretty much, or they're related to something. So, like you mentioned, there's complex diseases like autism. These are called polygenic diseases. There's hundreds of thousands of variants that are related to autism, and you could find one of them, and that's great. But there's many more that are related to autism, right? What happens is that these companies don't have the technology to look at basically if you look at a single variant, it seems like it's more simple and okay, here's this one variant, right? And this is what it means, right? But actually that if you look at the impact of that one variant, that functional variant, it's like 0.01% of the total impact in general, right? And even if the genetic impact is less than 1%. And you're talking about genetics might be anywhere from a third to 50%, whatever it is, right? Maybe 30 to 80%. We're talking about that one specific variant that you're looking at is a drop in the bucket. Really, it's literally a drop in the bucket, and it can't tell you anything, and therefore it can't predict anything either. We've looked at this research internally. We've tried to test models where we look at one variant, and you can't reproduce that if somebody has this variant. Basically, it's a coin flip. Whether let's say
Yeah.
you say we've got this gene for autism, we can take that variant, plug it into different databases like the UK Biobank, and see, let's test this variant, basically replicate the research. You can't replicate it. And the reason is because they're looking at a very specific population. There's a combination of data mining, and they're finding a variant that has some element to it, right? There's something to it, but it's just one piece of a 10,000 puzzle piece thing. So it's basically, so what we do, first of all, if you want to predict actually whether someone's more likely to have a condition, you have to do polygenic risk scoring. And there's some people may claim they're doing polygenic. First of all these functional companies that are doing genetics, they do not do polygenic risk scoring.
Yeah.
And that bugs me because they don't understand what they're doing is not legitimate. It's just cheap. I can build any of these products in a couple days with our team. Just here's the list of SNPs, here's the color and whatever based on the major, minor allele or whatever it is. It's not relevant, right? We actually offer that service. If any practitioner comes, they want to commit to a certain number of patients, we'll build you whatever PDF you want with whatever SNPs. It'll look like whatever PDF. We have all the data, all the SNPs. We have very good imputation, so we could take a 23andMe file and get all the variants that you would want from it, or even the ones that don't show up, because you could actually predict very well the other variant. So we can do all that, and it's just if somebody wants to do it, we can do it for them. But the idea is that we don't. Just to be very honest, it's not legitimate. We've tried to look at those models and see what can this predict. Can't predict anything. And I think it's helpful in maybe if somebody already has an issue. And the way we use specific variants is if somebody has an issue, it could be that if you have one variant or another, it might prioritize a certain recommendation. It might show you, okay, there could be something in this pathway that's a little off because we know you have this issue. And therefore, if you have, let's say there's many variants in the CBS. So let's say we'll put together like 20 variants, 5 variants, whatever it is, and give you a total CBS score, right? You could then say, first of all, it's not one variant, but you could then say, okay, based on that, there could be something. If you already identified that this person might have a CBS problem, you could see, okay, there's some genetic element here as well. That's something that you could do, right? But what we do is we look at we're looking at hundreds of thousands of SNPs to predict whether somebody is at risk for something or if they have a predisposition. And what I see is like people with glutamate issues, for example, they'll often have high risk for things like tics and anxiety, and like a bunch of glutamate type disorders. And sometimes if you have somebody with high levels of inflammation, general autoimmune issues, you'll see a bunch of them come up as a bunch of different autoimmune disease risks because it just shows you they have predispositions for different kinds of autoimmune issues, right? But that's very different than looking at single variants. So the whole looking at single variance thing is a money grab, in my opinion, meaning like it's very crude technology and it doesn't actually deliver any results except for some entertainment value. So what you need to do is really look at hundreds of thousands of variants, and then you need to validate that because you could say, hey, we look at 100,000 variants, a million variants. Okay, thank you very much. How do we know that's more accurate? You take these biobanks, these public data sets, and you can plug it in and see how predictive is this model. And you could put in whatever you want. If you have a single, what we found is these polygenic risk scores, when we plugged ours in, we found it was way more significant than these single variants and also more significant than a lot of these other polygenic risk scores. And then the algorithm takes into account also genetics, lab tests, symptoms, conditions, goals, lifestyle risks. Like it takes into account all the inputs. So if you upload lab data, it'll take into account all those inputs. And that's why I'm interested in organic acids. I want to see if we can incorporate those certain organic acids as, okay, somebody uploads their test and it could help improve the algorithm in terms of what they need to do in terms of lab testing. Because
Look, I think I can't see why that's not possible. And I think because all of these markers, if I just take the organic acid test, all of them involve enzymes, and as genes are what encodes for enzymes, right? So there'll definitely be patents there. I wouldn't know off the top of my head what a lot of them would be, but there would definitely be interesting patents there. And yeah, that sounds very exciting. And for me, I think that's where I would like it to go. So when I said that, I hope that people don't get too fixated on the genetics, especially the single polymorphisms. But if we can find a way to integrate that with tests and other things like that to really get a very complete three-dimensional picture, I think that's where we need to go. And I tend to agree with you. You mentioned something that's really been something that the old days, and you'd probably know where this was in the beginning with the nutrigenomics, it was like, oh, I've got an MTHFR mutation. What do I do with the MTHFR mutation, right? And what do you want to do with an MTHFR mutation? It's one SNP, right? One gene, one SNP. That does it mean something? Probably not. But that was a very trendy thing, right? It was the MTHFR bandwagon. Everyone got tested for MTHFR and looked like, yeah, but it's one gene from an ocean of genes. What does it matter?
yeah, the MTHFR is significant, but even though it's significant, it has
It's
to be
one.
like it's one gene, right? So you have to take it into account for in terms of many other things. So I could just show you, just for example, what that looked like here. So for example, if I look at MTHFR, you have people selling like, okay, you have MTHFR, here's exactly what you need to do. What this is showing is, first of all, with sophisticated polygenic risk scoring technology, we could see all the genetic predispositions and then have a best fit model with what fits in. That's my approach. And I think that's yours as well.
Very much. And so it's really for a practitioner, it's really good to see a company do that because as you mentioned, there's a lot of players out there and it, it's not necessarily that the information and the reports they put out there is useful. Like you said, it's like for maybe more for something else. So for me, it's really good to see companies do that because that's how it should be done. It should be having that holistic approach and not, oh, here's your DNA report. Now take a methylfolate supplement and then that'll fix all of your stuff, right? It needs to look at all those things. So that's really good, Joe. I'm very impressed with it.
Yeah, appreciate that. And I agree completely. It's exactly, you get a report here, you need to take this because you have this nip here. It's seriously, come on. You got this nip and right, like literally you're going to be taking 100 million things because you have 100 million variants. This one's
Sure.
more important. Okay, I got it. But still, the idea is that you can't operate that way just because maybe you could say, okay, look, I'm really just trying to figure out between A or B. And because you have MTHFR, let's go with folate instead of B12 to lower your whatever. That's as a theoretical case. That would make sense, right? I think me using this multi-dimensional approach actually has been a game changer for my health. Just like really like I started like following the algorithm and just one by one, I'm like, holy, like I just blind spots that I didn't see. just by optimizing, taking into account all the data, it had a huge game changing effect.
So what you're essentially doing there is you are waiting, you're looking at the trees and then you zoom out and then you look at the whole forest, right? That's what that program is doing, which is why it would
Correct. So,
work. Yeah, it doesn't surprise me that it worked for you because that's how you do get better, not by zooming in on the trees and taking a supplement for every tree, right? Been a pretty fun inspiration. I love the light part of it and the fact that you went into a whole bunch of rabbit holes. Really enjoyed it.
yeah, I appreciate you coming on, and yeah, we should hopefully stay in touch, and let me know if you're traveling at all. Maybe we'll bump into each other at a conference. And when I get my results back, I'm gonna be hitting you up and annoying you. What does this mean? What does that mean?
Absolutely. We'll analyze it and I'll know more about you than than I do now.
Thanks so much for coming on, and yeah, I appreciate the good work that you're doing. I feel like there's a lot of garbage in the functional medicine space, and you came highly recommended, and I could see why. You do know what you're talking about, so I appreciate that. Where can people find you?
Oh, yeah. So people can either go to my website, which is just elizmalambert.com, or they can contact us at office@elizmalambert.com. So that's definitely two avenues that they can get hold of me.
67% of listeners aren't subscribed to the show, so please don't forget to show your support by hitting the subscribe button now. You'll not only be supporting the show, but also investing in yourself and your health journey, all while helping to keep us ad-free.